This screencast video lecture explains about the factors that are affecting the bacterial growth there in the nature. Bacterial growth in nature is highly influenced by various types of environmental factors, as the bacteria are much sensitive towards the surroundings or habitat in which they are growing. These factors determine whether the bacteria can able to survive, metabolize, multiply, or they become dormant or they die in that environment. Basically, the requirements for bacterial growth can be visualized as a two-part. One is environmental factors that can able to influence the growth. Other one is a source of metabolic energy that is required for their survival and multiplication there in the environment. First, we try to understand the various environmental factors that could be affecting their growth. The first one is a nutrient, that is what type of nutrient have been available there in the environment. Based on that, the growth could be influenced. Here, nutrients are necessary elements that are required for the synthesis of a new cells, their reproduction or multiplication. In general, the following are the essential nutrients that are required for any kind of a microbial growth. They are divided into major elements, minerals, micro elements, and growth factors like that. Say major elements refers to carbon, oxygen, hydrogen and nitrogen that are required for the biosynthesis of carbohydrate, proteins, fats that are required for the cell. And the other minerals are sulfur and phosphorus, potassium. They are all coming under the other group of minerals that are essentially required. The next one is a trace element, are also called as the micro elements. They are playing a major role there in the enzyme functions are all. Example, zinc, iron, manganese. Then comes the growth factor. They are all basically of organic compound that are required in minute quantities for the microbial growth. These particular growth factors cannot be synthesized on their own by the microbe. So, it need to be provided or it need to be available in the environment. Then only it can be utilized. Thus, this kind of substances that are much essential for the growth of the organisms are supplied as a nutrient, for example amino acids. Next we look at the other factors, say how pH could affect. pH affects the enzyme activity, transport mechanism as well as the membrane integrity of the cells. Most of the bacterial cells will be preferring a neutral pH, that is of a 6.5 to 7.5 pH, whereas those that prefer to grow in an acidic pH, that is below 6, are referred as acidophils, whereas those that grows in a basic pH, that is more than 8, are referred as alkaliphils. Then most of the pathogenic bacteria that has the ability to grow only in a neutral pH, that is of a 7.2 to 7.4. An example is a Vibrio cholerae, which is a common pathogen there in the human or other mammals. It can grow in a pH of around 8.2 to 9. Next, we try to understand the role of certain gases there in the growth and survival of the bacteria there in the environment. Here comes the first one is the oxygen, and the another important gas is the carbon dioxide. If you look at the oxygen requirements of an organism, they can be broadly classified into aerobic, facultative anaerobic, anaerobes as well as microphils. Aerobes are those that cannot able to grow without oxygen. Very typical example is a Mycobacterium tuberculosis. And facultative anaerobes are those that can able to grow both under aerobic as well as anaerobic condition. They are commonly referred as a facultative anaerobic bacteria. Most of the members of Enterobacteriaceae such as E. coli, Shigella, Salmonella are all coming under the category of facultative anaerobes. The next one is anaerobe. They grow only in the absence of oxygen. Example are Clostridium and Bacteroides. The next and last category is the microaerophils that grow best in the oxygen that is of a lesser concentration. That is below 21% oxygen presence is the one which makes favorable for their growth. Very typical examples are Campylobacter as well as Azospirillum. Technically, if you look at aerobes and facultative anaerobes will have the following metabolic system. That is, they will have a cytochrome systems for the metabolism or managing the oxygen concentration that occurs there, and they will be having the superoxide dismutase as well as catalase which help to manage the free radicals or the hydrogen peroxide that could be generated during the aerobic growth of the organisms. Next, we try to look at the points related to carbon dioxide. Bacteria require carbon dioxide for their growth. Even though if they are of a heterotrophic organism, they also require the carbon dioxide. Whereas for an autotrophic organism, it is definitely required to fix their own carbon. Most bacteria produce carbon dioxide, that is at the end product of the metabolism. Finally, the carbon that have been taken inside will be metabolized, especially glucose taken inside is metabolized and they are finally converted into carbon dioxide there in the TCA cycle. So that is the process by which carbon dioxide will be leaving the system. Here, some special groups of bacteria which are referred by the term capnophils are able to grow better only in the presence of at least a 5% of carbon dioxide concentration. A typical example are certain pathogenic organisms such as Neisseria gonorrhoeae as well as Neisseria meningitide. Next, we try to look at how temperature can able to influence the bacterial growth in nature. Most bacteria prefer a mesophilic temperature, that is of a 20 to 45 degrees centigrade. Especially mesophilic bacteria will be growing in a 30 to 37 degrees centigrade. So most of the human pathogenic organisms will be falling under this category. That's the reason common pathogenic bacteria will be growing in a 37 degrees centigrade, which is again the body temperature. Based on the temperature requirements, there are lot of other categories. Say for example, psychrophils are those organisms that can able to grow at a temperature of 0 to 20 degrees centigrade. Whereas thermophils, which are commonly present there in the hot environments, can able to grow at a temperature of 45 to 80 degrees centigrades. Hyperthermophils are extreme heat loving organisms which can be growing in a temperature of more than 80 degrees centigrade. Next, we look at some points related to how water influences the growth and how ionic strength and osmotic pressure can able to influence the microbial growth. As we already know, water is essential for all the living organisms for their metabolic activities, mainly to take the nutrients inside. And technically, the water requirement for an organism can be measured as a water activity. Here, in dry conditions, when the water activity is very less, bacteria can able to convert their cells into a dormant forms which are referred as a spores. So, high water activity can able to favor the growth of the microorganism, whereas the low water activity can able to inhibit their growth. Next, we look at some points related to light. An optimum condition for growth of microorganism is only a darkness, as intense light may have a deleterious effects, especially the UV light can able to damage the DNA as well as inhibit the growth of the organisms. Whereas some organisms such as photosynthetic bacteria, for example cyanobacteria, may require light, but they can also able to grow under a dark environment by using heterotrophic nutrition mechanism. Then we look at some points related to the presence of inhibitory substances. Here they are collectively consists of antibiotics, heavy metals, toxins and disinfectants which can able to inhibit or kill the bacterial population. In order to counter effect this particular inhibitory substances, bacteria have developed various kind of resistance mechanism. For example, antibiotic resistance. Some enzymes like beta-lactamase expression could lead to cleaving the beta-lactam ring of the antibiotics. As a result, they can able to successfully compete with the other organism and live in the environment. And another classical example is the presence of certain efflux pumps there in the bacteria, which can able to send out the heavy metals or antibiotics out of the cells. Next, we look at the points related to competition with the other organism. As bacteria are growing in their natural environment, they need to compete for the nutrients as well as space with the other organism. This could be best influenced by production and secretion of some specific substances such as bacteriocides that can able to inhibit the other closely related competitors who have been present in that environment. So this is a table which shows the various factors that can able to influence or affect the growth of bacteria there in the nature. Apart from these various factors, the final one is a source of metabolic energy. This is also an important factor that helps in their survival in the environment or even in their growth and multiplication. Mainly three mechanisms have been understood about the generation of metabolic energy there in the microorganism. They are all fermentation, respiration, which consists of both aerobic and anaerobic respiration, and photosynthesis, which is a process by which the atmospheric carbon is getting fixed into cell carbon. Nutritionally, microbial growth could be effected based on the three important factors, that is the energy source. Here, the energy source can be obtained from sunlight. Those organisms are referred as a photosynthetic organism, whereas if the energy source is obtained from the chemical inorganic compound, they are referred as a chemosynthetic organism. Then carbon source. The next factor, carbon source, refers to how they are obtaining their carbon. Autotrophs obtain their carbon from the atmospheric carbon dioxide, whereas heterotrophs can able to use the preformed carbon. The third one is electron donors and acceptors. Who are all the molecules that are serving as an electron donor in the system and who are accepting the electrons finally in the respiratory pathway?
Based on the combination of energy, carbon source and electron donor, they can be broadly classified into four groups. They are photolithotrophs, example purple sulfur bacteria; hemolithotrophs, example nitrosomonas; photoorganotrophs, example purple non-sulfur bacteria such as rhodospirillum; and hemoorganotrophs involves various group of bacteria and fungi.